Welcome, everybody, to our seminar entitled Modeling Gut-Liver Interaction Using Epintestinal Microtissues in a Human Dynamic Multi-Organ Plate, HUDMOP. It's my pleasure to welcome you all to this webinar. My name is Jim McKim. I'm the president of IONTOX. IONTOX is a contract research laboratory that focuses on in vitro models, the application and development of these models, and they're used to understand the safety of drugs and chemicals. We have two excellent speakers today, and before we get started, I'd just like to give you a few housekeeping notes. First, the control panel that should be in your GoToMeeting panel allows you to ask questions. You can send those questions and we'll field those at the end of both talks. We'll answer any questions. It would also help us if you would mute. Be sure your microphones are muted. This helps a lot with feedback. Our first speaker is Dr. Seyum Ayouni. Dr. Ayouni received his PhD in medical microbiology from the Karolinska Institute in 1992. He then went on to do a postdoctoral fellowship at the Dana-Farber Cancer Institute at Harvard Medical School, and is currently the Vice President of Immunological Systems at MatTek Corporation. Over the last 4 years, he has developed a new in vitro primary human cell-based organotypic small intestinal tissue model that they refer to as EpiIntestinal. And he'll be talking to you about all the advantages of this system. Our second speaker, Dr. Jamin Willoughby, received his doctorate in molecular and cell biology from the University of California, Berkeley. Jamin has been working in the in vitro area for more than 10 years, where he's has gained a large amount of experience in the development and use of three-dimensional tissue models and their application for safety issues. And Dr. Willoughby currently serves as the Vice President of Toxicology at IONTOX. So without any further delays, I'd like to let Dr. Ayouni begin his presentation.
Thank you, Jim. And thank you, everybody, for joining us in this webinar. So 4 years in the drug development space, animal models have been utilized. However, for preclinical testing of safety and for drug safety and efficacy. However, they are costly and time consuming and also less predictive. And they are less translatable to human conditions. And more recently, there were a big push in emerging technologies that give emphasis on 3D tissues that utilize primary human cells, owing to the fact that it is more representative of tissue morphology.
Excuse me, Ayoum. Seyum, I'm sorry, we can't hear you. Could you make sure your mic is in the right position? Thank you.
Can you hear me?
Yes, not much better. Thank you.
Okay. There are now emerging technologies that give emphasis on 3D tissues that utilize primary human cells, owing to the fact that it is more representative of tissue morphology and predictive of drug responses in vivo. And to make sure these tissue models represent the in vivo-like tissue, they have to mirror the in vivo-like morphology and mimic the typical organ structure. And they have to also be more predictive of drug responses and disease in humans. And they can be, these models can provide results that can be translatable to human outcome, and they can be used in organ to organ co-culture systems using the microphysiological systems, which we are going to discuss in the second part of this talk. In today's talk, I will try to present, or I will go over the intestinal microtissues, reconstruction, characterization, and then I will touch with the different application of this tissue model in terms of drug permeation and metabolism, gastrointestinal toxicity, and restitution and inflammatory responses. And also, we'll talk about the adaptation of this type of tissue model to microphysiological formats. So our technology, this is a schematic representation of our technology. So what we do over here is we have a human organ donor, and then we get intestinal explants. From there, we can isolate epithelial cells and fibroblasts and expand them in culture in order to get enough number of cells to make thousands of tissues. Once we expand them, we cryopreserve the cells and then take them out and feed them onto insert membranes and then make them differentiate and stratified using optimal culture conditions and modified media, which has been developed at MatTek. And then while this optimized condition will give us a fully differentiated intestinal microtissues with a villi-like structure as shown here. So once we isolate the cells, we have to make sure that these cells are also epithelial cells and fibroblasts. So we did some preliminary, we did some staining to make sure that they are the right cells that we isolate. So here is the monolayer of epithelial cells in a phase contrast microscopy imaging, and then they express the tight junctions as stained by ZO1. And for fibroblasts, we used a different fibroblast marker, vimentin. So these red cells are vimentin positive fibroblasts. So the cells are epithelial cells and fibroblasts. And these monolayers, if we are going to use them for cultures, what they have to have some kind of stemness in some of the cells has to have stemness in their characteristic feature. So if if we have stem cells there, then they can repopulate the intestinal epithelium. So as you can see here, these are the monolayer cells, stand for the stem cell marker LGR5, and the red ones are LGR5 positive cells. So it has this population of cells that we isolate has stem cells in this population, and in some extent they also show a goblet cell marker MAC2 over here in red. So the cells are appropriate cells to use for the construction of the intestinal tissue model. So then I will take you to the schematic representation of the MatTek tissue models. As you can see here, these models are static systems grown on insert membrane. So we see the cells in here on the mic. We see the cells in here on a microporous membrane, and then that tissue culture, we grow them in what we call air-liquid interface to make sure that we enhance differentiation and the stratification of the tissue. And this type of tissue model will allow you to apply materials either topically from the top or basolaterally from underneath. So this is the schematic representation of the insert. Then we did a lot of characterization to make sure that these tissues are representative of the organs that we have in vivo. So here is the explant tissue with a villi-like structure. And here is the epintestinal tissue that we grow at MatTek Corporation. And these villi-like structures have villi-like structures have microvilli on the surface, and these microvilli cover when the microvilli cover the epithelial layer, they form a structure known as brush border, and that brush border formation by the explant tissue is shown as shown by electron microscopic imaging. You can see the brush border formation here on the villi-like structure. And then when we look at our own MatTek produced tissues, we can see very, very similar brush border formation. And this brush border formation is critical component of the small intestinal tissue model, because most of the drug metabolizing enzymes and the drug transporters are located in the brush border area. And the additional advantage of this microvilli-like structure on the surface of these cells is because this intestinal tissue is mostly absorptive organ, so they increase the surface area by about 600-fold. And we have published this result in Pharmacokinetic research in the April issue. So those of you who are interested in that, you can look at our publication. So unlike the organoids, the intestinal microtissues have an open luminal side, as you can see it over here. There is a protrusion of villi-like structure over here, and then the brush border is indicated by a special marker villain, which is green here. So you can see it is on the top, and then columnar epithelial cells are shown here by CK19 staining. So this type of tissue model will also, it is physiological, and it will allow you also to incorporate other type of cells, like the fibroblasts that you see, over here. So these are the fibroblasts, and this type of format will allow you to see a crosstalk between fibroblasts and epithelial cells. Other cells, as needed, you can make this type of format can make this tissue more complex. You can add additional cells like dendritic cells and so on. The full thickness tissue models that we have developed in the presence of fibroblasts can also de novo synthesize the extracellular matrix protein. And these extracellular matrix proteins are critical for growth, differentiation, and migration of cells. So in this case, you can see the green over here. You can see the collagen 4 expressed by the cells, by the tissue, and also fibronectin over here. And the red one over here are cytokeratin 19 expressing epithelial cells. This is a full, it has its own matrix protein, and this type of tissues are critical for, particularly if we are going to look at immune responses or innate immune responses in this type of tissue model, or if you want to study inflammation. To make this, the intestinal tissue model is also known for having subsets of cells. So these cells, so we have to characterize our tissue model if we are seeing these subsets of cells in our intestinal 3D tissue model. So what we can see here is in Vivo, you can see enterocytes, pennate cells, LGR5 stem cells, positive stem cells, M cells, Taft cells, goblet cells, and enteroendocrine cells. You can see here, for example, over here, you can see the enteroendocrine positive cells. Here the Taft cells and the MAP positive goblet cells over here, and the lysozyme positive pennate cells over here. So we think that our tissue model has appropriate cell types of the small intestine. So we continue characterization of the tissue by looking for if we are going to use it for drug permeation, drug absorption, and drug metabolism studies. We have to this tissue has to express drug transporters, both efflux and influx transporters. And in this case, as you can see here, the major drug efflux transporters. The major drug efflux transporters are shown here for PGP or P-glycoprotein, MDR1, and then the top panel is our tissue model, and then the bottom one is explant tissue. So they express the efflux transporters in the similar manner to the explant tissues. The next question is do we have the right polarization of this tissue model? And to see that if the transporters are located at the right location, we just did a preliminary simple experiment over here. So what we have seen. Sorry. So what we have seen, what we are looking at here is what we can see here is the villain on the explant tissue, and then on the second panel, on the second part, you can see the PGP expressed on the apical surface, and then the overlay on the bottom one, you can see the overlay of PGP and villain. So meaning the P-glycoprotein is located on the apical surface. And on the right-hand side, we have the human intestinal tissue models that we developed over here, and they have exactly similar localization of the P-glycoprotein on the brush border location. So the next question is, how long we can culture this tissue model? If we want to do long-term experiments or chronic type of experiments, then the tissue should be able to grow for a certain time period. So we did some experiments to see how long can culture this tissue model with when keeping the physiological condition. As you can see it from here, because most drugs are dissolved in DMSO, we use DMSO as a vehicle control to look at the tissue membrane integrity, if the integrity of the tissue is intact for a long period of time. So as you can see, you can grow this tissue for up to 6 weeks. This allows you to do more repeat dose recovery experiments. So I will take you to the application part now. So one of the applications is drug absorption, and this has been published in the Pharmacokinetic Research in the April issue. The more details can be found there. And I will give you the summary of what we have published. And what we did was we looked at the gold standard for drug absorption studies since the 1970s is CACO2 cells. How is the performance of our tissue model when you compare it with CACO2 cells in terms of drug absorption and drug permeation? So we took about 11 drugs, known 11 drugs from the different categories, and then test them both on our tissue model and on CACO2 cells, and compare them with the percent fraction absorbed in humans from the literature. So when we do that, we can see here that for our tissue model, the correlation was, the R-squared value was about 0.1, whereas for CACO2 cells, it was the correlation was about 0.71. So that shows you that we have improved the improved correlation of human absorption, drug absorption in the epintestinal tissue model compared to the CACO2 cells. So our model is similar to that of the in vivo responses. So the next one is, if we are going to use this tissue for a long period of, for a certain time, then we have to have reproducibility of the tissue. So we took 4 representative drugs, and we look for the P-up values of 3 different lots of productions. And as you can see, the intestinal microtissue shows excellent lot-to-lot reproducibility. Then we look also nowadays, a lot of people are taking a lot of drugs at the same time. So we were looking for drug-drug interaction in this model. And as you can see, efflux transport, as in here, for example, this is the Tylenol is the substrate, and verapam is the inhibitor. So you can see that when you add the inhibitor, you can increase the bioavailability of the drug while you are decreasing the efflux ratio. So these are the key points of this slide. So you can increase bioavailability by adding these inhibitors. So the tissue model can be used for drug-drug interaction. And then the activity of the metabolic enzymes that are expressed on this tissue model, like CYP2C9, CYP2C19, CYP3A4, and CYP3A5, were also analyzed using isozyme-specific fluorogenic substrates. And as you can see, there is activity of the tissue showed some activity in terms of metabolizing substrates. And then to make sure that it is functional, we also use ketoconazole as inhibitor of CYP3A4 to look at if the metabolism activity is reduced. So as you can see in the second, in the bottom part, you can see the inhibitor were able to reduce the activity. So the other application of we have GI toxicity application on this drug model, and we are collaborating with companies on that, and we are going to publish that paper. And we have seen that drugs that have been failed in humans has been predicted correctly in our tissue model. So I'm not going to touch that one, but here there is another use is for inflammatory studies, for example, and this tissue model has to recover itself. And as you can see here, we damage the tissue, and then we followed it through time. And as you can see here, by day 4, the epithelial cells, which are cytokeratin 19, the pink cells, were growing into the healing, into the wounded area, and the fibroblasts were covering. And by day 7, there was a full healing of the wounded area. And you can follow this type of wound recovery by transepithelial electrical measurement, and by day 7, it basically when it fully recovers, it just comes to the normal, the physiological level. And you can follow by immunohistochemistry, by histology, as you can see over here. By day 3, a single layer of cells were there, and then you can see stratification of cells by day 4, and a complete reepithelialization of the tissue by day 6. So you can follow it by different methods. And the other application is the host-microbial interaction, and this we have published it in Get Microbes. So you can follow. I'll show you some of the bacterial infections that we did on this model. So as you can see here, this is E. coli infection. The green is E. coli, and when you infect the tissue with E. coli, they are only attached to the villi-like structure or the cells. And then when you go to the pathogenic microorganisms, that is Shigella, you can see after 4 hours, the red ones are Shigella invasion of the tissue. So you can see here inside the cells. So the tissue can be used for infection experiments, and this is good for the microbiome. Could be good for microbiome research. In general, after infection of the tissue, you can also induce bacteria. These bacterial, live bacterial infections can induce innate or inflammatory responses. And in this case, you can see that interferon beta here, and TNF-alpha and IL-1 beta, all of them are upregulated by the wild type, but not by the commensal bacterial infections. You can study immune response, innate immune responses tissue model. And in conclusion, this tissue is can be has a morphological structure like the human intestine, and the primary intestinal they can be used for different applications, and they can be cultured for up to 6 weeks. So the applications are Cytotoxicity, drug absorption, drug-drug interaction, intestinal restitution, pathogen host interaction, and inflammation. At the end, we have I have addressed the use of intestinal tissue as a standalone organ model. However, there is a compelling need for more physiological relevant systems for organ crosstalk in preclinical and pharmacology and toxicology. In the next part of the presentation, you will hear about organ crosstalk using microphysiological human dynamic multi-organ plate. Thank you.
Okay, while we're changing over, our next speaker will be Dr. Jamin Willoughby.
Well, thank you, Sayum, for that exceptional talk on the Epithelial model and your MatTek's in-depth characterization of the Epithelial model. We really like this model. And thank you, Jim, for the introduction. As he said, my name is Dr. Jamin Willoughby, Vice President of Toxicology here at IONTOX, and I'm going to discuss how we're using this Epithelial model in an integrated in vitro organ platform to evaluate cholestasis, which is a pretty big problem in the clinic. So we're going to give a little background on cholestasis, discuss bile acid homeostasis, and as well as a quick discussion on the adverse outcome pathway for cholestatic toxicity in the liver. And then we'll discuss IONTOX's Hudemont platform. It's our optimal in vitro intestine and liver model, which we used here, and how we use that model to evaluate cholestatic processes in an in vitro integrated organ model. So first, we're going to start out by asking the question, what is cholestatic liver injury? Well, cholestatic injury and mixed cholestatic hepatocellular injury represents over half of all drug-induced liver toxicities in the clinic. So this is a pretty important problem that pretty much all pharmaceutical companies are facing when they're developing drugs. And it really comes about by dysfunction in bile acid synthesis, transport, or signaling pathways that control the homeostasis of the bile acids. And this can be either acute or chronic. It can be mixed hepatocellular with cholestatic injury, or it could be biliary. It's typically characterized by a disrupted bile flow, which leads to pure intrahepatic cholestasis, and of course, obstructive cholangiopathy with injury in the bile duct epithelium. To understand bile acid homeostasis, cholesterol here is metabolized by CYP7A1. This is actually the rate-limiting step in bile acid synthesis. And in particular interest to this presentation, we're looking at enterohepatic circulation. So the bile acids are exported out of the liver partly through the bile salt export pump, BSEP here. They're then after ingestion of a meal. They're then exported into the intestine, where they can then be taken back up into the body, into the liver through ASBT, which is the apical sodium bile acid transporter. And so there's a lot of control mechanisms for bile acid homeostasis. So hepatic signaling mechanisms that control bile acid concentrations are pretty significant. There's a lot of control mechanisms in place. And these are controlled primarily through the farnesoid X receptor, or FXR receptor. So when the FXR receptor is activated, it leads to an increase in FGF19, suppression of CYP7A1, which again is the rate-limiting step of bile acid synthesis, and induction of BSEP, or bile salt export pump, the MDR3, and OST alpha beta, which is a heterodimer and is a bile acid efflux transporter. And so obviously there's a number of ways that the drug can impact these pathways. And these red boxes here are highly dependent on FXR, and this was published by Jackson et al. From Qualis BioIBT. And this response is known as the adaptive response, and there's synergistic effects to this adaptive response. And so the response can be initiated by BSEP inhibition. So here on this graph, we're looking at OST beta, which again is one of the heterodimers for the bile salt efflux transporter. We're looking at gene expression here on this axis, and when we apply cyclosporin A to the liver, cyclosporin A is a BSEP inhibitor, we get an increase of OST in order to compensate for the lack of BSEP being able to export the bile salts out of the cell. When we vastly increase the intracellular concentrations of bile acid, we see a similar response. So when we either block BSEP or increase exogenous CDCA, we get a response from the OST beta. However, when we do both at the same time, so we're inhibiting BSEP as well as increasing intracellular concentrations of CDCA, we get this synergistic effect and this massive increase in OST beta mRNA expression. And so this is what is known as the adaptive response. And this is all in an adverse outcome pathway. This pathway is pretty well defined. It's well known. It's actually an OECD approved pathway. And basically, just by looking at this, you can see that cholestasis is a really complex process. There's multiple mechanisms involved in the regulation of bile acid concentrations. And the current approaches have really focused on BSEP inhibition as alone as a predictor for cholestasis. But clearly, there's a number of other pathways that can be targeted. So it's much more complex than just BSEP inhibition. And so for our model, looking at cholestasis, we use human primary cells. Typically, we can also use rat primary hepatocytes. And these are done in sandwich culture, and these are obtained transporter certified hepatocytes obtained from Qualis BioIBT. And the reason why we use the transporter certified primary hepatocytes in sandwich culture is because they form bile pockets. They also have essentially the full complement of uptake and efflux transporters, not only present but functional, and the FXR signaling pathway is intact. Although we are looking at other possible models in the system that may work, such as liver spheroids and certain clones of the HepaRG cell line. And so again, we're using these primary hepatocytes from Qualis BioIBT. They're transporter certified, so they have the full complement of transporters here. And in sandwich culture, they form tight junctions and also these bile pockets. And so the localization and function of the uptake and efflux transporters is really crucial for fully understanding cholestasis, as well as extrapolating from in vitro to in vivo. And so again, the other model is the Epithelial model. So we had to characterize this model as well. Again, we're looking at enterohepatic circulation. So the presence of ASBT is really important. And here in this graph, we did some qPCR on the Epithelial model. And we're looking at just the threshold value of that qPCR. And what essentially the take home from this is that in the presence of both cyclosporin A and triglydzone, these are known BSEP inhibitors, as well as co-exposures with cyclo and the bile acid CDCA, or triglydzone and the bile acid CDCA, there's not much of a change in ASBT, although it is present. And of course, we just have GAPDH here as our normalization gene. And again, here, when we look at the FXR related genes, FGF19, CYP7A1, and ASBT, there's essentially no change in the intestine when we look at these. We can also inhibit the activity of ASBT. This is done using a 500 micromolar single exposure of fluvastatin in 50 microliter dose. And what we see is about a 30 to 35% decrease in the uptake of GCDA. So we can actually inhibit the activity. So it's present and functional. And so our experimental hypothesis is that an integrated organ model can not only recapitulate single organ responses, but can add important information on drug kinetics, bioavailability, and inter-organ metabolism, which would improve the predictions of systemic effects in vivo. The drugs that we chose to do this work, again, were cyclosporin A and triglydzone. They're known to disrupt bile acid homeostasis in vivo. Cyclosporin A is an immunosuppressant that's really widely used, and it's a really potent BSEP inhibitor. And liver toxicity due to bile acid increases in the liver is really quite rare with this drug. Whereas with triglydzone, it's highly associated with liver toxicity. Although it is a drug for the treatment of type 2 diabetes, it is a BSEP inhibitor, and actually it's an FXR antagonist, both the parent triglydzone itself and its metabolites. And so this compound is known to cause DILI in the clinic, which is cholestatic drug-induced liver injury. And the physical and chemical properties of these compounds can change. So here we have triglydzone and some of its metabolites, and then here's cyclosporin and a couple of its metabolites. And if you look at some of the physiochemical properties, we have a lower molecular weight, a pretty high C-log P, but a low polar surface area. For triglydzone, whereas cyclosporin has a high molecular weight, about the same C-log P with a much higher polar surface area. And so just looking at the physiochemical properties, you'd think that triglydzone would be a much better drug and a much safer drug, but we know that that's not really true. And this is because the Lipinski rule of 5 would suggest that this would not be that good of a drug. But in fact, because we know that it is, we know that these physiochemical properties are altered. So here's our experimental design. Our Hudemop setup we have in our first chamber here would be the intestinal tissue. That would be in this sort of photo, that would be set up probably here. And then we have our human hepatocytes and sandwich culture. That would be in this cup. And you can see this green line here. This is actually a semi-permeable membrane that flows through. Inside the cup here, it's permeable. And outside of these cups, there's a non-permeable region here. And this actually simulates blood flow. So we can perfuse through a buffer that works by essentially diffusion kinetics, and it mimics blood flow through the system. And so we applied a single dose of test material to the apical surface here, 30 micromolar cyclosporin, 300 micromolar triglydzone. And then 6 hours later, we spiked in CDCA at 30 micromolar here in the liver, and that's just to increase intracellular CDCA levels. And then we collected intestinal media, now the basolateral here, the liver media here, as well as this simulated blood, this what we call the perfusate that would then come out at this end of the system. And then we assessed the tissues for cytotoxicity and gene expression. And one of the key components to this setup and to our Hudemop platform is that there's no direct mixing of the media. With most all other multi-organ methods out there, there's a singular media that must be used and optimized for all of the tissues involved on the plate, whereas with our setup here, we can have each tissue in its optimal media because there's no mixing of the media. So here's a couple more photos of the setup. And here we would have, so this is our pump set up with a syringe, pushing the perfusate through. Here we have an epiintestinal tissue, so the perfusate goes through here into the intestine, and here we have a third cup, which could be kidney or any other organ of interest, and then we would collect the perfusate here. And what this allows us to do is to sample all the major compartments within a tissue. So for example, we apply our drug, we can look at drug permeability, parent permeability, we can look at metabolism by the epiintestinal, we can look at absorption in through the semi-permeable membrane, followed by delivery of parent as well as the initial metabolite into the liver, and then look at the metabolites that the liver is then producing, either by metabolizing the primary metabolite or the parent itself, and then we can collect this out in the perfusate. So by doing this, we can look at intracellular fluids by looking at the tissue, the liver tissue itself, the epiintestinal tissue. We can look at the extracellular fluid, which would be the media, and then even the plasma or blood, which in this case is mimicked by our perfusate. And so when we look at the uptake of the drugs in the basolateral compartment of the intestine, what we can see is with both triglycerin and cyclosporin, we see a pretty rapid uptake of the material into the basolateral region. We're getting about 58% permeability with triglycerin and about 90% with cyclosporin. And this actually is really close to the published literature on the bioavailability of these drugs. And then when we look at what's in the liver model, what we noticed is that the drugs were at or below the limit of detection. So the development of highly sensitive methods is really key to our kinetic data. But when we look, what we see is a lag phase before we begin to see accumulation into the liver compartment. And this lag phase is due to the time it takes for the parent to get through the intestine and then into the perfusate and then out into the liver. And this is also due to the fact that once it is delivered to the liver compartment, the liver cells will be rapidly taking up and metabolizing both triglycerin and cyclosporin. And so we see that nicely represented here with this lag phase. So then we looked at whether or not the adaptive response in the liver was activated or not in our system. And what we see is bile acid alone or drugs, cyclosporin and triglycerin alone, had no effect on the fold induction of the genes we looked at. So we were looking at the FXR-related genes, FGF19, CYP7A1, OST alpha, and OST beta. And this black dotted line here is a twofold induction, which is what is considered to be a biologically relevant induction of genes in a qPCR system. When we look at cyclosporin, which is a BSEP inhibitor, we get a huge increase in FGS19, a big decrease in CYP7A1, a little increase in OST alpha, and then a really significant increase in OST beta, up to 40-fold plus. Now, when we look at in the presence of triglycerin, which is not only a BSEP inhibitor, but it's also an FXR antagonist, we should see a reduction in the adaptive response, because yes, we're inhibiting BSEP, but then we're also inhibiting somewhat that adaptive response mediated by FXR. And we see that here with about a 20% decrease in FGF19 and about a 50% decrease in expression of OST beta. So the adaptive response is definitely reduced by triglycerin. And so we're seeing clear effects of this at really low concentrations. If you remember here, we're looking at nanomolar concentrations. And this could be significant for risk assessment of pharmaceuticals, because we're observing these responses at concentrations much lower than those observed when you're just adding drugs into a liver compartment in a typical in vitro study. And so the next question is, well, did we actually see any cytotoxicity associated with this? And yes, we did. So when we looked at LDH released in the liver compartment, if control, we didn't see any. Cyclosporin, none. Cyclosporin plus CDCA, none. We saw a little bit with triglycerin, 1% to 2%. But when we look at triglycerin at 300 micromolar plus CDCA at 30 micromolar, we see about a 17% increase in LDH released. Now, this is not a huge increase, but it is statistically significant compared to all other conditions. But not only that, we're seeing this hepatocellular toxicity observed at a really low concentration, at a nanomolar concentration. So, and this was also published by Jackson et al. This figure, but when we look at hepatocellular injury, so in a typical system, we have bile acids coming in, and the intracellular bile acid concentrations are regulated by BSEP. Here it's exporting into the bile pocket. When we block BSEP, we're no longer able to export. We're still getting import, and the concentration goes up. This then leads to an FXR activation, which causes an increase in OST alpha beta, and then we get a subsequent decrease back to stable homeostatic levels of bile acids. However, if we block FSR antagonism, we then get the subsequent increase, and if we also block, or we can also block basolateral efflux by blocking OST alpha beta or MDR3 or 4, then we get a huge increase, which can lead to liver toxicity. And so compounds can essentially increase the intracellular concentration of bile acid through BSEP inhibition plus some other form of inhibition of other bile acid efflux transporters and or by inhibiting FXR antagonism. And so what new information can we learn from this integrated organ systems? Well, we can assess permeability. As we've shown, the bioavailability in the literature or the bioavailability that we're seeing here with triglydzone and cyclosporin is really close to the levels that are in the published literature. We can assess first pass metabolism, not only in the intestine, but also in the liver. We can look at enterohepatic circulation, which we could do, and is something that we've currently been modeling, especially since we know we have the ASBT gene present in the Epithelial model. Another thing we've learned is that the target organ dose is much more accurate as the perfusate, the simulated blood system, works on diffusion kinetics. And what we're seeing is a much lower target organ dose, but this actually closely mimics the in vivo situation. And what this then allows us to do is we can really model the PBPK and so in conclusion, we have a well-characterized integrated organ system which provides valuable kinetic and exposure data. And this test system that we used here demonstrated that the key hepatic intracellular signaling pathways controlling the bile acid homeostasis are functioning, and they are responding properly to the cyclosporin and the triglydzone exposures. Our ASBT, the apical bile acid transporter, is functioning in the MatTek Epithelial model, and the bile acid toxicity was observed, but only with triglydzone, which is what is known to happen in the clinic as well. Triglydzone is a cholestatic drug-induced liver injury positive compound. And so these data reveal that target organ doses are much lower than when directly applied, which more accurately mimics in vivo situations. And as I said, PBPK models are possible with this type of data. And so we'd like to acknowledge the lab staff here that worked really hard on this. Nick Hibbert, our Director of Lab Operations, and Bonnie Corpus, who's our research scientist. And of course, I'd like to thank MatTek for their help with the Epithelial tissues as well as with this webinar. So thank you, and I guess time for questions and answers now.
Great, thanks, Jamin, and thank you, Seyum. There are several questions out there. A couple for Seyum first. Can you talk about the DMSO concentrations that you've used in the Epithelial? Maybe also talk about the maximum concentration.
Hello?
Yep, go ahead. Seyum, are you there?
Yes, I am here.
Can you answer the question?
Hello?
Yes, can you?
It's the DMSO concentration was less than 0.5%.
Less than 0.5. And another question for you, Sayum, is does the PAP change when you're doing the apparent permeability using the Epithelial? Does the PAP change with the age of the tissues?
The whole experiment in this case was done at we manufacture the tissue, and then it was done within 3 days of the culture period. So after that, so but we have looked at in the 6 weeks period, we looked at the different characteristics of the tissue, and we didn't see any significant change over there. But we didn't do at different time points, we didn't do any drug permeation studies.
And Jamin, so a couple of questions for you. One is the types of tissues that can be used in the plate platform and the maximum number of tissues.
So target the first question, what types of tissues? We have worked with a number, essentially anything. We've looked at lung, as far as 3D goes, we've looked at lung, we've looked at the Epithelial. We could use ocular tissues, 3D tissues, dermal 3D tissues. As far as what's in the plate, we've looked at the primary hepatocytes. Obviously, we're in the process of doing quite a bit of work with some kidney, hypothalamus. Basically, anything that can be cultured in a 6-well plate can be cultured in this system. And what was the second question?
Just the number of tissues that could be. Yeah.
So, right. So we've gone up to 3. So we have the application tissue and then 2 downstream. We can link up to probably about 6 tissues. The problem is that as this works with diffusion kinetics, the drug gets more dilute as it goes down. So we just have to ensure that the dose that we apply to the first tissue isn't so cytotoxic that it essentially kills the tissue. But we can go up to 6.
Okay, so another question. They want to know if you're using the Epithelial full thickness versus just the epidermal layers. Can you do PAP in both models?
Yes, you can do on both models. There is no difference in terms of application of the drug. But the only difference between the 2 tissue models is one is more complex and the other one is only epithelial tissue. So except that the application is both, it will allow you to do both apical and basolateral applications.
Okay. Jamin, there's a question regarding the medias and talking a little bit about how the media is not mixed.
Right. So the let's see. Is my screen still up with the Q&A?
Yes.
All right. Let me go back to our photos here. So what we have here in this syringe is a buffered system. Now this buffered system essentially mimics human blood. Now as this is on a timer, on a pump, and it'll pump the stimulated blood through the system. And if you can see here, this portion is non-permeable. In here it's permeable. So anything at about 30,000 Daltons or less is essentially diffusible into this simulated blood flow. So the parent that we apply and this tissue will then go into the basolateral media and then be carried into this cup where, again, by diffusion kinetics, it diffuses out into the compartment over time, and then metabolites and or parent can then also diffuse back into this system, be carried into the next cup, and so on. So each tissue is in its optimal media, and parent compound and metabolites and things like that are carried through the system through this semi-permeable membrane.
Yeah. Yeah, and just to add to that, there's an osmotic balance there. So only the molecules that are osmotically active will actually move between the simulated blood and the chamber. So because all the buffers are osmotically balanced, there's no net change, there's no net fluid change, no net volume change, chamber to chamber. Okay, I'm just waiting for some more questions. For Sayum, in terms of determining the tissue barrier integrity for the PAP experiments, can you talk about how you do that? Are you using tear measurements or using a leakage marker?
We use leakage markers. So with every drug, luciferase was added topically. So that's how we see it. And if the luciferase leakage after drug treatment is less than 4%, then the tissue is intact. But if it is over 4% of luciferase leakage, then that indicates that the drug has some effect on the tissue. So that means there should be adjustment of the concentration of the drug.
Okay.
So in all cases, luciferase was used as the leakage marker in all drug absorption experiments.
Okay. Jamin, there's a question. They want to know if we've ever used spheroids in the plate, and if so, what were the results, or can you just speculate on how they might work?
So the answer is not yet. That is something that we're definitely getting into. There's a number of potential benefits to using spheroids. I think that the primary issue with using spheroids is, and one of the benefits to this model is that these cups are about a 6-well size, and what that allows us to do is to analyze multiple endpoints. So we can look at viability through LDH release, we can look at viability in the tissue, we can look at gene expression in the tissue. And when you're working with spheroids, unless we have a whole lot of spheroids in that cup, it kind of limits the amount of endpoints that we can do. So no, we haven't. Yes, it's something that we want to try, and it's something we want to get into. We're kind of looking at how many spheroids per cup are we getting. Are we getting the same amount of metabolism that we are with the primaries and things like that.
And the same person wants also to know what the business model is for the IONTOX service. Which is basically, they develop a protocol. We develop a protocol with you. You send us the compound. We run it in our plate system per the protocol that we develop with you, and then send you the data.
Right.
And then Sayum, a question to you about applying particles to the tissues. Can you do it apically? Well, obviously you can do it apically, but has anyone done it, or have you done it basolaterally?
Yeah, when we do A to B and B to A, the drugs are for B to A. The drugs are added from the basolateral side. For A to B transport, the drugs are added on the apical side.
Okay, and what about toxicity studies? Have you done it? Have you looked at the effects on the tissues from the basolateral exposure?
Yeah, it has been done. So we have looked at it. Yes.
Okay. Well, we're about 5 minutes past our time, and we really appreciate everyone's attendance. And if you have any additional questions, please contact Sayum at MatTek Corporation or Jamin Willoughby at IONTOX. So again, thank you all very much, and this concludes our webinar.
Thank you very much.